the best advice and mentorship I feel like I've gotten has been through peer mentorship. So with, colleagues that are just a few years ahead and, vice versa, right? So like with us all together, we have a similar shared experience. And we can learn from each other. So you can learn from someone else's mistakes, right? Before you make the same.
Welcome to another episode of the Beckman Bites podcast, organized by the Arnold and Mabel Beckman Foundation. Here, we'll be diving deep into the research and career interests of Beckman Scholars, Arnold Beckman Postdoctoral Fellows, and Beckman Young Investigator Awardees. This podcast should be interesting to students and researchers of all varieties. We'll discuss various research experiences, different scientific fields, advice for undergraduates and graduate students, and much more. This video will be made available on YouTube and the Beckman website, so if you know someone interested in science or the Beckman Foundation, please be sure to share this video with them. Today, I'm chatting with Professor Ming Hammond. Professor Hammond has been part of the Beckman family for some time now. She was named a Beckman Scholar in 1998 during her undergraduate degree in chemistry at Caltech. She obtained a PhD at the University of California, Berkeley, studying peptide mimics to treat amyloid diseases. She is now an associate professor of chemistry at the University of Utah. Her research group develops new bioimaging agents for studying metabolites and signals in bacterial cells, and has made various discoveries in cyclic dinucleotide signaling. Thank you so much for being here, Professor Hammond.
Hi, Zoheb.
So can you give us a little bit of your background and your career path?
Yeah, sure. Well, so I'm currently an associate professor in the Department of Chemistry and also a member of the Henry Eyring Center for Cell and Genome Science here at University of Utah. And before that, as you kind of already discussed, I went through a trajectory going from the East Coast to the West Coast and back to the East Coast. And so now I'm here in Utah and Salt Lake City.
Great. How are you liking it there compared to Berkeley? Is the weather as good?
Well, I think skiing is very good, and in the winter months, so we're coming upon ski season right now, so it's very convenient, maybe 45 minutes to get to the slopes. But also, I grew up in a pretty flat place, so I'm still learning to ski, but it's really lovely to be hiking in the mountains during the summertime too. We have a lot of national parks, you may have heard.
Yeah, that's really great. Can you give a little bit about your current day-to-day tasks being an associate professor and running a research lab and teaching classes?
Yeah, so I think that I remember thinking when I was an undergrad and also as a grad student, right, that it's really what professors do in their offices is really like this black box that I really didn't understand. So what I wanted to say about kind of day to day is that overall, it's kind of very much a choose your own adventure, right? So yes, one of the major parts of my job, which I really enjoy, is running my research lab. And so being an academic lab, we're equally focused on both pursuing scientific questions that we're really interested in finding answers to, as well as training students to do research at all levels. And then, of course, we also do teaching, which includes kind of formal teaching. So I've taught everything from general chemistry to organic chemistry lab to graduate courses in chemical biology. And then the choose your own adventure part is really there's a lot of work that professors do in service to their departments and also to the greater scientific community. So right now I for the lab, I am, helping with a student who's proofing their manuscript. I'm writing a grant right now, but also for the overall scientific community, I'm co-editing a journal issue.
Wow.
Graduate admissions is coming up, so we're gearing up for, reviewing all the applications coming in. And also, I'm serving on an organizing committee for a national conference in synthetic biology. So, it's just a little bit of a snippet of other things that you do besides research and teaching.
Yeah, that sounds like a lot of different things on your plate. Is it, you kind of have to, some days you're probably going pretty heavy at manuscript writing, and then some days it's all about teaching classes and grading?
Yeah, I think it, just as I remember as an undergrad, kind of juggling all your courses and your homework load and your labs and so on. It's the same kind of juggling that you have to do. But I think one thing that I found, which I think is true for a lot of people, that there's certain times of day where I'm just the most able to be the most productive, and like my mental capacity and ATP levels are highest. So in the mornings, I usually reserve that for writing and research. And then I try to fill in, the whole rest of the day gets kind of filled in with other things, but try to reserve kind of this precious early morning time for the writing.
Wow. And I mean, I'm sure doing all of these things is kind of what you love to do, being a professor. Does it ever get, I mean, I'm sure it gets exhausting to be juggling all these different things. Um, but it's kind of crazy to me that professors can handle all of these different things. I mean, organizing a conference just sounds like a complete logistical nightmare.
Yeah, well, actually, like I said, I think it's very much a choose your own adventure. So, it's often a privilege and an honor to be asked to do some of these things. And it's sometimes hard but important to have to say no to some of the opportunities that you get. So yeah, in this case, these are various conferences and journal topics and things that I really care about. So yeah, I find it to be rather worthwhile.
Yeah, that's really amazing. Can you tell us a little bit about how you kind of got to this position and what educational steps you had to take, and a little bit about how you merged into being a professor?
Yeah, so I think um, I feel like I had a pretty standard route, but with a change in topics, perhaps. So, as you mentioned, I got my bachelor's in chemistry, and during that time was when I first became, I guess, a member of the Beckman family. So, as a Beckman scholar, I was in Barbara Imperiali's lab, and it was a really, I think that was the experience that during my undergraduate career really solidified my interest and intention to go into academic research. And then I went on to do my PhD at UC Berkeley in bioorganic chemistry, as you alluded to. And, I think that was a time where I really got a lot of experiences that actually broadened my experience into teaching, for example, and other opportunities I got during my PhD. One somewhat unexpected opportunity I had was that I was actually Paul Bartlett, my PhD advisor's last graduate student. So he announced his intention to retire in my second year of graduate school. So that meant that I at a certain point became the most senior member of the lab and the only member of the lab. And so it really kind of forced me to become a lot more independent and to start to, kind of reach out and kind of build my own scientific community and, kind of cohort, since the lab kind of shrank in front of me. And then after that, I actually did a postdoc and switched fields from chemistry to RNA biology, which is what really catalyzed the change to the research that I do now in nucleic acids.
It's really interesting that you mentioned the end of your PhD being kind of isolated in your lab. Did you have to reach out to people in other labs doing similar peptides for amyloid diseases and trying to collaborate in that way?
Yeah, so I was pretty fortunate that one, the project that I worked on was quite collaborative. So I was the synthetic chemist developing a new class of peptide mimics to target amyloid protein. And we're really fortunate that Professor Stan Prusner's lab is, Nobel Prize winner, who discovered these the amyloid proteins, was at UCSF. And so we collaborated with him and also collaborated with Wendell Lim, who now is one of the forefront leaders in synthetic biology. And so through my research project, I was able to, have interactions with scientists, not just at Berkeley, but also at these other institutions. So I thought that was really helpful. But then, within my own lab, as I said, I think I spent maybe 6 months literally being the only person in the lab, maybe 3 months. But what, I quickly determined was that I started attending other research group meetings because I wanted to kind of continue to learn new chemistries, and, participated in their group meetings. And it just really kind of forced me, who's a very introverted person by nature, to kind of come out of my shell to continue to kind of connect with people. Definitely.
Yeah, that's kind of amazing that you managed to keep those connections alive and strengthen them. And I'm glad to see it's strengthened your career and allowed you to have more meaningful scientific collaborations too.
Yeah, I think actually was really one of the things that was really beneficial was I actually talked with Carolyn Bertozzi, who was on my thesis committee when I was a graduate student, and she had kind of a similar, not exactly the same scenario, but she had a similar situation happen to her in graduate school. And I remember she was just so passionate about saying like, this is no problem. You're going to be fine. This is what I did to, kind of keep the lab spirit alive and to kind of pull through. And just she had so much belief that this was possible, right? And she was like kind of proof of that. And so I think I just remember that was a conversation that really kind of, helped me like reset and say like, okay, yeah, this is not a problem. I can do this.
Yeah, that's amazing that you turned what seemed to be a disadvantage into an actual blessing, since you forced you to collaborate more and reach out.
Yeah, exactly. I think right. I think it's just how you view things, right? So there's always kind of a silver lining to it, I feel, right? So there's definitely, some negative aspects, but on the whole, like I said, you can kind of say, well, what can I do about the situation, right?
Right, absolutely. So let's dive a little bit deeper into the bioimaging agents you study and signaling in bacterial cells. What kind of motivated you to enter that field coming from peptide mimics?
Yeah, well, it very much was driven by the fact that I had an opportunity to attend the Bioorganic Gordon conference when I was kind of midway through my PhD. And during that conference, I heard the person that became my postdoc advisor give just an amazing talk about the discovery of riboswitch RNAs, which are these incredible biomolecules that are able to toggle genes on and off in response to chemical signals. And it really, that kind of talk, and then, kind of looking into the literature, really opened my eyes to the fact that nucleic acids are not just, involved in the code of life, which is, of course, really important, but they're also really sophisticated, machines. They can do sensing, they can be part of programmable enzymes, and then they also can play really important roles in signaling. So, after that time, I, resolved to really learn as much as I could about RNA. So I did a postdoc in Ron Breaker's lab at Yale, and it was always with this intention that I had to, eventually think about combining, kind of new knowledge that I would gain about RNA with kind of my chemistry and synthetic chemistry background. And, what kind of came out of that was this research program where we're interested in using or making nucleic acids into molecular tools, right, for imaging, for detection, and possibly engineering and therapeutics. So, this research program that, I've been running for several years now really spans this area that's now called chemical biology, also spans synthetic biology and kind of classic biochemistry fields as well.
Yeah, it's really neat that you've made your way over from short proteins to nucleic acid. Now you just have to cover lipids and sugars, and you've hit all of them too.
Yeah, well, I think maybe the guiding thread, if there is one, is that I've always been interested in molecular recognition. Mm-hmm. So that's just been a passion of mine. So when you're trying to oftentimes when you're trying to design a drug, like we were with our peptide mimics, to break apart a protein aggregate or to target some enzyme, you're trying to make a non-covalent interaction that's specific to your target, right? And so, yeah, that's molecular recognition in a nutshell. And I've always been interested in kind of how can we exploit molecular recognition for all sorts of different applications. And probably in my mind, one of the most challenging applications is getting two things to come together and make a specific interaction in a living cell with all the other proteins, other nucleic acids, and also other small molecules present, right?
Yeah, that's brilliant. So what's it like mentoring undergraduates who might just be, they might have just taken organic chemistry or biochemistry, and you're trying to get them to do very high level, cutting edge research?
Well, I think I have had many students start in the lab as freshmen. And like you said, they haven't taken almost any college level biology or chemistry classes. But what I find is really, and also I should say that I started in my lab as a freshman, right? So when I was an undergrad researcher. So what I find is true is that it's so much more empowering to get the knowledge that you need as you're learning to do something, right? So, I find that, the subject matter comes to life because you need to do a PCR reaction. Well, let's learn about how DNA gets synthesized, right? So it's just, you're directly applying the knowledge. It's so much easier for a student to learn that in the context of being in a research lab. So so that's why I say that kind of running my research lab and doing research with students undergrads, grad students, postdocs, and the like. All of that is kind of part of the teaching mission of a research university.
Yeah. I mean, I'm sure your students are very motivated to memorize the 20 amino acids and know the side chains perfectly. So I'm sure if you could go back to your undergraduate years, you would tell yourself certain things. What are some things that you find yourself telling your undergraduate students the most about research career and surviving undergraduate?
Yeah. Yeah, so I was just kind of thinking to the what we were talking about a little earlier about, I was thinking about what I tell my undergraduate mentees, the students that are majoring in chemical biology and come to me to kind of talk about questions about their schedules and things like that. One of the pieces of advice that I give students, especially students that are actually in research labs, I usually ask them, have you talked to your advisor this semester, right? So, I think it's surprising to me that, but it's oftentimes, right, students, they work with graduate students or with postdocs on their research projects, and they're really, have the same feeling that they have with maybe professors that they see in their classes. They don't feel very comfortable talking with the professor, even though they're in their lab, right?
Mm-hmm.
And so I would actually make it a point to tell my advisees, then before you see me next semester, you need to have at least one conversation, because oftentimes the questions they're asking me as their advisor are questions that I think also, maybe they're related to graduate school, maybe they're related to, research opportunities and things. I said, these are great questions, and here's what I think. But I think you should also ask your research advisor, right? It's just a great, you're working in their lab and, kind of putting in your blood, sweat, and tears into research in the lab. And I think, all of us that have undergrads in the lab, I think we really appreciate that, right? And so, I think we would be happy to talk with students, but you sometimes have to reach out, right? Or, make the point to make an appointment and just sit and talk for 10 minutes, right? It doesn't have to be very long.
Yeah, there's always that barrier you have to break between graduate students and postdocs, and at some point it's got to be demystified that there are these completely different people that are just incredible. I mean, I love talking to undergraduates and sharing the advice that I have, because I feel like I know so much about how to kind of navigate an undergraduate career. So it's kind of shocking that undergraduates hold back questions when graduate students and postdocs love talking about that stuff.
Yeah, well, there definitely are shy graduate students as well, right? So I think, like I was explaining that, it really took almost like a shock to the system to kind of force me to come out of my shell and be a lot more outgoing and, proactive about maintaining contacts with people. And I think the main thing is to say that just try it. It's an experiment, right? So that's what I usually tell students, try it one time, see how it goes, right? With different faculty, because you may not hit it off with everyone, right? There just might be some curmudgeons in our department, there usually are some, but you might find someone that could become your mentor, right? And they don't necessarily have to be your research mentor either, right? And I think, all of us have had some experience, maybe even before college and so on, of someone that was helpful, right, in a meaningful way. And it's like, wow, if you maybe want to, it's hard to find those kinds of people, and it doesn't necessarily happen the very first time you talk with someone, right? But if you, again, just are open, you can and experiment. Like I said, you might find people that you really resonate with, and they can be helpful.
Absolutely. You never know who your magical mentor will be that really shows you the way and helps you out the most.
Well, I think that's another thing I wanted to say. I tell this to actually my junior faculty colleagues these days, which is that also, this is from a class actually for faculty, but I think it's very true across the board, which is that you don't necessarily need or even want there to be just one person in your life that you rely on for all knowledge. In other words, someone is your research mentor, but they're not some kind of magical guru who knows all and can be everything to you, right? So I think in fact, in my own experience, I've had a lot of some of the best advice and mentorship I feel like I've gotten has been through peer mentorship. So with, colleagues that are just a few years ahead and, vice versa, right? So like with us all together, we have a similar shared experience. And we can learn from each other. So you can learn from someone else's mistakes, right, before you make the same. And so I think peer mentorship is another really very, very powerful tool. And I think for whether we're talking about undergraduates, graduate students, or junior faculty, I highly recommend it.
Were there some unique learning opportunities that you had during your PhD experience and becoming a faculty member?
Yeah, I think that, the thing I was thinking about is that actually in each stage of my career, I feel like I was incredibly fortunate to have had a chance to kind of try out my wings in terms of exploring independent research, right? So for the Beckman Scholar Program, I think that was really the first time, right? I got to propose doing something that was a new project, that was my own project that I would get to start in the lab. And then in my PhD, as I already mentioned, I kind of really had to learn to become independent in my own lab of one, right? So I think in some ways it kind of prepared me for learning how to start a lab in some sense, but not all. And then during my postdoc, I was really fortunate that I received a career award from the Burroughs Wellcome Fund. And so that allowed me to spend time at Heidelberg University and pick up some skills and training in plant genetics of all things. And that was something that was independently funded by the Career Award, which was really valuable.
Yeah, that's really neat that you kind of got that extra learning opportunity.
Yeah, so it's the training in plant genetics, is very far afield to what the lab does now. But actually, there's a side benefit to that. There's a long trajectory that connects that first experience at Heidelberg University. And I think I mentioned to you, we recently launched a synthetic biology experiment to the International Space Station in December of 2018. And it was a plant synthetic biology experiment that traces back all the way to that first experience that I had a chance to do during my postdoc.
Wow, that's pretty incredible. I don't think a lot of scientists get to say they're doing an experiment in space.
Yeah, it was an incredible opportunity. So one of the, I think it was one of the experiments where, I got to take my family, including my now 7-year-old son, to see the launch. So it was, one of those special moments when you can say you're watching a rocket take off and you know that something that you designed is inside. That's incredible.
Wow, that's a really cool experience.
Yeah.
What were you trying to study by sending a plant in space?
Yeah, so we wanted to test whether or not we could um, produce a protein, so activate the expression of a protein, a transgenic protein in a plant.
Well, good luck with it getting submitted. I think that'll be a pretty interesting paper to read.
Yeah, thank you.
Yeah. Um, can you talk a little bit about how the Beckman Foundation has influenced you through being in the family and such?
Yeah, so like I said, I think probably the major thing for being a Beckman Scholar for me was that first opportunity to try out, proposing my own research project, having to defend it in an interview was something that I don't know what was your experience, but that was something that I had to do. And also, my undergraduate research lab actually moved across the country from Caltech to MIT during my time as a Beckman Scholar. And so one of the things that, because I had, this independent funding and it was on this project, I was able to essentially go to Cambridge, Massachusetts, and that was my, and finish my research project there. And so it was kind of my first time kind of going off on my own and, navigating a completely different city for a summer, and all the meantime kind of helping to get the lab set up and seeing what that was like. And also finishing my research project. So
Yeah,
yeah,
sure.
that's
I'm sure your
really,
advisor really appreciated the help with transitioning and such. I know transitioning labs must be a nightmare.
well, again, there was, graduate students and postdocs in the lab that were, of course, setting everything up. And actually, my undergraduate research advisor was incredibly kind. She had said, well, give us, that you have these 10 weeks, um, and the summer is 15 weeks long. Why don't you wait 5 weeks before you come so that hopefully we'll be set up by the time you arrive. So she actually, wanted to have everything ready so that I could finish my project actually. So, but it was still a really uh, transformative experience getting to do that as an undergrad.
Yeah, that's really fantastic. I definitely feel pretty similarly. Just the experiences of
Mm-hmm.
for the Beckman Scholarship and going through that interview, like you said, and then all the resources. It really just pushes you to have so many more scientific experiences and really learn about what you're doing and sink your teeth more into it.
Yeah, no, and I remember, again, I was in the inaugural class, so I think there might be a lot of things that have changed in the program since when I started. But I do remember that, at the end, we had a chance to do a symposium at the Beckman Foundation. And actually, Dr. Beckman was still alive at the time. And so, we had a chance to meet him at the symposium. So that was really incredible too.
Yeah, that's really neat. Did he give advice to you directly, and what was that experience like?
Well, I think, we had a chance to kind of meet him all together, all the Beckman scholars from various institutions. I don't think I had a, kind of one-on-one time with Dr. Beckman. I guess I should have been a little bit more proactive, like I was saying, but I wasn't at the time. I was pretty shy, right? So didn't have a chance, to get a personal advice from him. But
Were you mainly So I imagine with molecular recognition, you're interested in binding constants with your peptide, with your um, with the systems you were looking at, and characterizing it in different ways.
Yeah, so we did have to develop assays to measure the affinities of our various mimics that we were designing. I guess to explain it in a little bit more detail, we had a hypothesis that, we have the standard amino acids, and we had the hypothesis that if you could make basically cyclic amino acids, so you can make all the versions for all 20 amino acids, but they're cyclic, you could constrain, so you would constrain what conformations the amino acid can undertake. And so in some ways, if you made peptide mimics that had these constrained amino acids, we could prepay the entropic cost for binding. So in other words, by making them somewhat rigid, we could make it so that they're set up to bind to specific proteins, with the amino acid sequence kind of dictating what proteins they would target. And so I was synthesizing, or figuring out how to synthesize these cyclic amino acids in the first place, and then how to make them into peptides using different solid phase and solution phase methods. And then once we had the chemistry all figured out, I mentioned that we collaborated with colleagues at UCSF who were studying protein-protein interactions. And so we designed some of these mimics to target their protein-protein interaction. And so I would drive over the bridge to San Francisco, carrying my samples with me. And, we would do the experiments over there to make the protein. And that's, kind of learning all of that. And then doing the fluorescence, the fluorimeter experiments to look at the binding. And it was definitely very tense every time, because I remember, thinking about it. You spend many, many years making these compounds and you have no idea what their activity is. And then, the moment of truth time, when you're about to inject the sample into your solution with your protein, and, you're watching the fluorescence levels change, and can't wait to go home and calculate out and see whether or not your theory actually, came into reality, right?
Yeah, that's really neat. I mean, I came from in my undergraduate, we worked on selectively recognizing peptides with a synthetic host molecule called cucurbituril, and I always got the same feeling too when I was doing studies as well. It's really neat that you were able to design those systems and tweak them to really solve problems in that area.
Yeah.
How has your perception of science changed since your PhD, now that you're a professor?
Yeah, well, I think now, I'm like behind the curtain, I guess. So I can see, how the sausage is made, right? So I would say that as an undergraduate, I was just so, I think as many students, you're just so enthusiastic about science, and you have a lot of idealism, right? And I think I'm actually still pretty idealistic, but I think also I would say that I've gotten maybe a little better at explaining my science to my parents and to other people. So that's definitely a skill that I had to learn with time.
Yeah, reaching broader audiences is very important for scientific legislation and just overall community knowing what's going on. That seems like a very important thing to know how to do.
Yeah, well, it's just but I think that it really is helpful to just, even as you're doing undergraduate research, learning kind of more about, well, I'm doing this research. It's part of, because I'm part of this group that's studying this thing. But it's definitely helpful to sometimes ask about the bigger picture, right? So that you understand the bigger picture behind the work that you're doing. It's just so helpful, because it helps you start to understand the context of like, well, what makes an experiment worth doing? What makes an experiment worth funding? Or not just experiment, but a series of experiments as part of research, right?
Do you encourage your students to give talks to non-scientists and?
Oh, yeah, we actually, I think maybe a year or two ago, we actually had what we called the Project Pitch Slam event, just for the lab. And it was just actually each of the undergrads worked with their graduate students and gave kind of a 5-minute elevator pitch on their projects, to each other and just to us. And everybody just, for every pitch we would clap very enthusiastically. But I think we awarded bragging rights really to the student that kind of gave the best pitch. But I thought it was actually really fun. The graduate students and the undergrads working together to come up with like a very broad audience description of what they're doing.
That's really neat, especially for students interested in going into entrepreneurial science or other areas. It just makes so much sense to be able to speak clearly about your goals and articulate that.
Yeah, definitely.
Is there any sort of advice you would give to yourself if you could go back to graduate school?
Well, so I don't know if this is advice that, well, I think this is something that I did as a graduate student that I thought was really helpful, and advice that I actually got from one of my mentors, right, when I was in graduate school, was kind of to keep practicing thinking and asking questions about science, like even while you're learning about science. So it's something that I think I'm pretty well known for asking questions during research seminars. So I find that it's really helpful to stay engaged and curious and open to learning about science outside of the very specific science that we do in the group. And it also really, I think, is a just broadly useful skill for research. For a researcher, this initial kind of asking questions about science, this is kind of what allows you to build your creative research muscles, right? In terms of thinking creatively about research problems as you're learning about it, like what are some alternative strategies that this person could do? In some ways, it's kind of working your way towards, coming up with and questioning your own research ideas, right? So you can ask questions of other people's research ideas, then you get practice at asking questions about your own research ideas. And that's how you kind of iterate and formulate and hopefully improve on your research ideas with time, right? So that was advice that I had gotten as a graduate student, and is something that I definitely try to pass on to my graduate students, is to kind of practice asking questions.
And that, while you were saying that, it made me think about how much more interdisciplinary science is becoming, with scientists from different fields coming together and solving interdisciplinary problems. So the problem solving that would be applied to engineering is now kind of being applied to more chemistry problems and vice versa. And I guess everyone's getting better at asking questions and approaching how to solve them.
Yeah, I think, maybe the one challenge, right, for those, for the interdisciplinary type research is really, bridging that gap. There's a lot of opportunities, and so I think that's kind of what catalyzed me, moving to a different field, was I just saw there was so much opportunity to bring the mindset or the strategies from a completely different field to apply to a new one. But it also can be an obstacle in some senses as well, because sometimes people only have a set way of doing things, and they might not be as welcoming of new approaches, or you might kind of have multiple homes in different fields, and that sometimes might feel like you have no home, right? So, interdisciplinary research, I think a lot of funding agencies, everyone thinks that this is incredibly worthwhile to give also cross-disciplinary training to our students. But then, what it really takes are people kind of advocating for each other's fields and saying, well, this is what, one field brings to enrich the other, and, helping to really strengthen those ties.
In general, how do you kind of handle, I know we're running a little bit low on time, so this is my last question.
Okay.
In general, how do you handle work-life balance? Does it exist being a professor? Do you help out your undergraduate and graduate students with this as well?
Yeah, well, I think there's a motto from my college days, which is work hard, play hard, right? So I think, what another professor that I met at a conference recently, the way she described it is that the balance might be on the average, but in terms of a day to day, it's impossible to have kind of whatever split you're planning for. Let's say it's 70-30. It's pretty impossible to maintain 70-30 throughout the day. It's more like during the course of a week, things kind of go up and down in terms of work and life, and so on and so forth. One thing I would say is that as an academic, I feel incredibly fortunate in terms of having flexibility in my schedule, right? This is, for good or bad, this is not a 9-to-5 job in that sense, right? You have an incredible amount of flexibility that a lot of people don't have. So I feel actually incredibly fortunate that, I'm able to make accommodations in my schedule. I make my own schedule in a lot of ways. And so that actually really helps with the balance, but the balance is not, keeping things constant. It's more kind of like you're adjusting on the fly for whatever is the most, pressing deadlines and other things that come up both in work and in life, right?
Yeah, absolutely. It sounds like you've really made the most out of that and really gotten to take advantage of that flexibility. Well, Professor Hammond, it's been a pleasure speaking with you about all these different topics. I think a lot of the students will really like and enjoy hearing about all your diverse experiences and the advice that you've given.
Thank you.
Before we end, I would like to thank the Beckman Foundation again for giving this platform. And if you watch this video and you thought it would be useful to someone, please go ahead and share it with someone you know that's interested in science. These sort of conversations with professors don't happen every day, and all of this advice is really quite a gold mine, especially the work-life balance. Knowing flexibility is really useful. All right, well, thanks for being with us, Professor Hammond.
Thank you.